everybody knows that within our cells we have DNA, which is basically the instruction manual for our cells to do their jobs. We have cells in our skin that are supposed to protect us from the outside. We have cells in our lungs that are supposed to help us breathe. Every single one of the cells in our body has exactly the same DNA, but in each cell they use it in a different way. And that same DNA is also the instruction manual for that very first zygote, the very first cell in the embryo to create a human being. And so it's really important to keep that information intact. We want to study really how cells preserve this information. Now, why do they need to preserve it? Because we live our lives in an environment that contains many agents that can harm our DNA. And when our DNA gets damaged, it can eventually cause the sequence of it to change. When a sequence of the DNA changes, that's what we call mutations. Mutations essentially are what make us different. That's why the newborn baby is different from its parents, because there have been some changes in its DNA. But also, sometimes they are problematic because the cells in our body have wonderful DNA encoding for everything they need to do. But if they accumulate mutations, they lose some of their function, and eventually that's what causes cancer. So what we like to study in my lab is to understand how these damages form in our DNA, and then we have diseases and cancer and things we would prefer not to. Nowadays, we have these amazing technologies that we can actually sequence the entire genome in an experiment. So we have 3 billion bases that compose the human genome that are composed of only 4 letters, A, C, T, and G. And so in my lab, we have technologies that allow us to take cells in culture. We grow cancer cells in a plate, and then we treat them with things that we know cause damage. Like we have UV irradiation that we can do in the lab, create damages in the cells, and then we can sequence the genome and we can say exactly where the damages occur. And then we have technologies that we can go through the genome and see what areas get repaired more efficiently, what areas get repaired less efficiently, and then try to understand when do these mutations occur. Today, we know that these mechanisms that the cell uses to repair DNA are super important. How do we know that? Because we know that when they go wrong, when cells don't have these mechanisms, that's when you see bad things happen. So the more we learn about how these repair mechanisms work, we're getting better and better at finding the people at risk and helping them find cancer early. The other thing where I think we would like our research to go, is that once we understand how these systems work, we can maybe try to manipulate them, maybe learn what's wrong in the cancer cells, and then target the cancer cells for therapy specifically. There are a lot of other places we could look at, and when we do research in the lab, we don't just work with cancer cells, we work with many cells, and there are a lot of places where actually DNA damages do good. For instance, in our immune system, there is actually an interest for the cells to be different. So there's a mechanism in immune cells to change constantly. So one of the ways they do that is by purposely changing the sequence of DNA, and they do that with special enzymes that specifically will create DNA damages to create mutations inside the immune cell. And so there are other places where we could start studying these things, but for now, I'm fascinated by mutations and cancer, and that's where we've been going.